get us started. I am so pleased to have Dr. Beckman with us today. She is a very dedicated physician scientist, and she is specifically dedicated to those with benign hematology conditions, so our favorite type of dedicated physician scientist. She has a clinical interest in acquired and congenital bleeding disorders, as well as those with anemia, and she conducts translational research in molecular and cellular mechanisms that drive inflammation-induced pathology in endothelial cells and blood vessels, which I actually think is the key to truly understanding the damage caused by sickle cell disease. So I am pleased to have her here today to share her research, and the floor is yours.
All right, well, thank you for the invitation. I always like to start with acknowledgements. I think we get rushed at the end and we put this up there and we don't really say anything, but I stand on the shoulders of giants. I have Dr. Belcher, Dr. Versalati, and Dr. Hebel, who've really sort of, way back when I was a wee med student, MD PhD student, they got me started on this path. And the little lab family that we have with Fuad, Chen, Julia, Ping, and then Dr. Kaiser, who's now, he's been the ASH Congressional Fellow and has moved on, but was part of the family when I rejoined the group. We are nothing without our summer students. We have a strong summer program here. And we had 2 students that contributed to the work I'm going to be talking about today. And then I've had some minor luck with funding in different areas. Full disclosure, my Novartis project is on crizanlizumab, but it's for myeloproliferative neoplasms and not related to sickle cell. So like I said, at Minnesota, we actually have, I was sitting down and preparing this talk just to familiarize with everyone what we have going on here. So we've historically always had a red cell vascular endothelium group, Robert Hebel, Greg Versalati, John Belcher, and I've been fortunate enough to sort of start to step up and join their ranks as a junior PI. We have a growing interest in gene cellular therapies with base editing led by some of our pediatric colleagues, Bo Weather, Brandon Moriarty, and Dr. McIver. We have an established pain neuroscience group with Dawn Simone, who's collaborated closely with Calta Gupno when she was here, Arena and Virginia Siebold. And then we have a biomechanical translational group. I've been working very closely with David Wood, and then our colleague Yvonne Dada and Alex Forscher. We've been working on developing the sort of translational aspect and looking more at the actual bedside aspect of the stuff that we've been doing in the lab. So sort of putting all this together, what are we going to talk today? So we're going to talk about vascular inflammation and sickle cell disease and how toll-like receptor 4 and heme really play a role in driving that. We'll be covering why hematopoietic versus endothelial cell toll-like receptor is important. And then sort of we're going to close with some strategies to prevent the toll-like receptor 4 mediated activation that we see in patients. So vascular inflammation, I sort of take a vascular or an endothelial centric view of the world. So I put the endothelial cell, I guess the blood cells are in the center. And it has a dynamic function, and it's one of the largest organs in the body besides the skin. It has an antithrombotic function, anticoagulant function, it participates in angiogenesis, wound repair, vasodilation, and it's a barrier anti-inflammatory function. And when this is really dysfunctional, you can see aspects of this. And I think we really see this in sickle cell. We see issues in all of these domains. So when we think about inflammation, we can have activation of complement, which I'm not going to talk about today, leukocyte adhesion molecules, which we'll get into in more detail, and amplification of cytokine storm through some other molecules that are on the surface. And with coagulation, we have some overlap. So again, we have adhesion molecules, so P-selectin, von Willebrand disease, fibrinogen, platelets. We also have cytokines that lead to platelet production, and we have tissue factor, which activates coagulation. So I don't think this group needs a big introduction. Some of my slide decks have huge introductions to sickle cell and the history. I didn't feel like your group needed that, but I really do consider it the original molecular disease. As we're talking about more personalized medicine, sickle cell has always been one of the first, and for many years, one of the only ones that was really studied. And so single point mutation causes a change in our hemoglobin S polymerization, but it really leads to global dysfunction of the vascular system. In the United States, it's a very under-recognized disorder in that it occurs in 1 in 360 African-American births. Trait is even more common, and that isn't even the tip of the iceberg in terms of the global burden of this disease. Now, I think it's like the 40th anniversary, actually 41st anniversary of this paper from Dr. Hebel and Marty Steinberg. describing the adherence of erythrocytes to the endothelium in sickle cell disease and how it was actually correlated with clinical severity. And I think that this is a paper that is one of those benchmarks as far as how we've taken to approaching sickle cell disease as a vascular disease, not just hemoglobin related diseases. It's not just the red cells. And this is a framework that I think we published this as part of a review, but it was definitely in my slide deck every talk I gave in grad school, where you have activated leukocytes and the sickle red cells interacting, releasing free hemoglobin, leading to reactive oxygen species, NF-kappa B activation, endothelial adhesion molecules, leukocyte adhesion, vaso-occlusion, and reperfusion, which then feeds forward into a vicious cycle. When you simplify it a little bit more, what you have is in individuals who don't carry the hemoglobin S gene, you have blood cells flowing across an endothelium, and they may stick to an adhesion molecule here or there, but it really doesn't happen all that often. When you have sickled red blood cells and that release of heme, you get increased upregulation of adhesion molecules, which causes some adhesion and some rolling. And if the insult is large enough, you eventually get occlusion, which decreases that flow rate and causes hypoxia or ischemia. And then when you have reperfusion, that cycle amplifies and you get upregulation of that adhesion and dysregulation of the endothelium. So how does toll-like receptor 4 play a role, and what does it have to do with heme? So the innate immune system is present in our body, and I sort of, when I describe it to patients, I say it's the dumb immune system. It's there to recognize patterns. And we know that LPS is a pattern that the toll-like receptor 4 will remember, and then it will signal through toll-like receptor 4 either in a MYD88 dependent manner, which then converges on NF-kappa B, or an NYD88 independent manner, which converges on STAT1. And so you have LPS binding to the receptor, leading to downstream signaling. In 2007, a group out of Brazil was one of the first to show that heme-induced TNF-alpha secretion in macrophages. And this was in both mouse macrophages and in human macrophages. And I think this is important to realize because the mouse immune system is not equivalent to the human immune system, and there's some slight differences that can play a role, but this seemed to be sort of a universal thing. When they looked closer at what could be happening here, they looked at toll-like receptor 4 knockout mice as well as MYD88 knockout mice. And there's no bars here because there was no TNF-alpha activation when you knocked out toll-like receptor 4 or MYD88, either in the presence of heme or. So what this says is that in the present, heme really signals through toll-like receptor 4 and through MYD88 dependent mechanisms in macrophages to activate cytokine release. So again, we're sort of showing this here in that TNF-alpha secretion from macrophages is dependent on toll-like receptor 4. What was really interesting is that they then went and blocked the LPS binding site on the toll-like receptor 4 molecule and really showed that heme signals independent of that. So heme had a totally different binding site than LPS, but is still able to signal through toll-like receptor 4. And so back into cartoon form, so we have LPS here, and this is an older review that has LPS binding protein. Well, we would probably say this is maybe heme, and it's binding to toll-like receptor 4 and going through this MYD88 pathway, which converges on NF-kappa B. So we wanted to know, where is heme interacting to activate this NYD88 dependent toll-like receptor 4 signaling? And so we know that LPS binds to this cofactor MD2. And so we were wondering if MD2 might be also cofactor for other things, such as heme. And so we asked, is there a heme binding site on MD2? So there's, luckily for us, computational programs that you can look and enter heme proteins into. And when we do these computational analyses, there's 2 potential heme binding sites on MD2. And a lot of proteins have heme binding sites. They tend to be rich in cysteines and other moieties. But in this case, there was some tryptophan, tyrosine, serines, again, some cysteines that were present on this structure. And so using molecular techniques, we made a luciferase reporter activity for MD2 activity and then mutated each of those potential domains. And what this bar graph is showing here is that when we did the magenta cluster, which contained some of these threonines and tryptophans, that we were able to, when they were mutated, sort of ablate MD2 activity. The green cluster actually had like a hyper response. And this was specific to heme. LPS didn't activate this MD2 cluster at all. So we were really having a heme specific response. We then took these mutants and we bound them to heme columns to see if they would bind to heme. And when the MD2 wild type is present, you have heme binding. When you have these mutants, and this in particular, the combination of all three mutants, you really get ablation of that MD2 heme interaction, and you don't have binding to the heme. And so we can now say that in particular, these tryptophan and this tyrosine motifs are the ones that are required for this heme activation of toll-like receptor 4 on MD2. And so putting this together, we know that heme can activate toll-like receptor 4 MYD88 signaling through interactions with MD2 to stimulate pro-inflammatory and adhesive pathways in macrophages. And we know that sickle cell has increased intravascular and extravascular hemolysis because we have heme released from free hemoglobin as well as free heme. And so going back to our sort of vicious cycle, if you come up here, we have a sickle red cell, which is hemolyzing and releasing heme or hemoglobin. And you get not only reactive oxygen species, but toll-like receptor 4 activation. Now, what we wanted to look at is do we see these downstream effects in our sickle cell animals? So Dr. Ghosh and company from the University of Pittsburgh, I think they might have been at Emory at this time too, they actually published in 2013, a little bit ahead of our group, that infusion of free heme induced death in sickle cell, but not trait or AA mice. So again, sort of extravascular hemolysis model with giving a large bolus of heme. And what they did show in this paper through a series of experiments is that if you quenched the effect of the free heme, either by using hemopexin, which has a high affinity for that free hemoglobin, or you inhibited toll-like receptor 4 with a drug called TAC242, and here they use 2 different doses, you could protect the sickle cell animals from mortality related to big bolus of heme. When they looked at the lungs in particular, they saw that there was some protection from acute lung injury. So here's a mouse lung that just infused saline. With heme, you get a lot of congestion, a lot of leukocytes infiltrating the lung parenchyma. You also get edema in the sort of airways, alveolar wall thickening, and hemorrhage. Now, when you gave these animals TAC242, you protected them. And they did one other experiment where they took sickle cell bone marrow. They radio treated toll-like receptor 4 mice. And so then they made a chimera where there was sickle cell red cells and white cells present, but that the vasculature didn't have toll-like receptor 4. And they saw that there was also some modem of protection against lung injury in those animals. Our group, about a year later, followed up, was showing that hemin triggers several things. One is Weibel Piloty body release. So Weibel Piloty bodies sit on the endothelial surface, and when they're activated either by histamine or other mediators, they secrete out P-selectin, von Willebrand factor, and other mediators to sort of activate coagulation and other signaling pathways. And we showed that hemin was a potent stimulator of both P-selectin as well as von Willebrand factor. And then when we look in the vasculature of organs, so this is a black 6 animal up here, and this is a strain of sickle cell mouse called the New York mouse. When we gave a bolus of hemin or histamine, we would get activation of von Willebrand factor and P-selectin in the vasculature in both animals about the same amount. And then in cultures, we can take endothelial cells from humans. Human umbilical vein endothelial cells are a common model that we use for this. And what we can look at is activation of NF-κB, that downstream pathway of the MYD88. We see as LPS can activate that. TNF actually does not signal through toll-like receptor 4. It has its own receptor. And so that does activate phospho NF-κB. And if you give heme plus the toll-like receptor 4 inhibitor, you block this. We can also do this in mouse lung pulmonary vein endothelial cells. So we can culture these. And again, we see that heme activates phospho NF-kappa B, LPS does, and TNF, and that's ablated with the addition of the toll-like receptor 4 inhibitor. And then over here, we actually isolated these pulmonary vein endothelial cells from mice that are knocked out for toll-like receptor 4, and we lose the effect of heme and LPS, but again, because toll-like TNF signals through a different pathway, we don't lose that effect. So we know that heme can activate NF-kappa B through toll-like receptor 4 interactions. So what does this mean for our animals, or for what our patients in the blood vessels are seeing? And so we can do a vaso-occlusion model where we can visualize blood vessels, and we can add different things into the system and see if we see vaso-occlusion. So in a sickle cell mouse, if we just give them an IV infusion of saline, we see about 10% of the vessels, give or take, that are occluded. If we add in just free hemoglobin, we get a greater increase, about 30% stasis at 1 hour in these vessels. And likewise, if we give free heme, and then in this experiment, we actually gave a bolus of water to induce intervascular hemolysis, and we get a similar effect, that we get stasis in about 30 to 40% of the vessels. Now, if we prevent the effects of free heme, so if we quench that with haptoglobin or hemopexin, we reduce that stasis back down to baseline. So we also then looked at the endothelial cells to see if those Weibel-Palade bodies are affected. And if we block toll-like receptor 4, we don't see any heme release of von Willebrand factor or P-selectin from the endothelial cell surface. And then in this experiment, what we did was we took, again, these are new ARC mice, and we gave them free heme, and we get stasis at about 26% of the vessels. If we give LPS, it's even greater, it's about 45%. If we give a control IgG, we, again, don't really affect that result very much. And down here is our control animals that if they see no sickle cell, they still have a little bit of stasis, about 5%. And this down here is hypoxia, so that is ischemic insult independent to the heme. So if we block the heme effect with TAC242, we reduce that stasis down to almost baseline. If we block the LPS response with TAC242, we also block the stasis response. And if we block it with an anti-LPS antibody, we get that same response. And the same thing if we block the anti-LPS antibody with heme, we actually don't have as much of a response. So, because again, heme signals independent of the LPS binding site in our animals. And then with hypoxia, we also see that release. So this sort of indicates that free heme signals independently of the LPS binding site in our animals to mediate vaso-occlusion. And so if we think about this in sort of a schematic, visual learners, you have hemolysis releasing this heme. It can go maybe to the vessel wall, but it could also potentially be going to these monocytes and macrophages and activating cytokine release, which then activates the endothelium. And so we didn't really know, is the heme present on the vascular wall, or is it the heme activating the macrophages that's really causing this effect? And so the next question we wanted to address was, is the heme-mediated effect of toll-like receptor 4 dependent on leukocytes or in the vasculature? And one of the reasons why this question wasn't really answered by the previous studies is because bone marrow chimeras tend to have toll-like receptor 4 on their hematopoietic cells. So if you take an SS toll-like receptor 4 animal, and then you transplant that bone marrow into a toll-like receptor 4 knockout animal, what you get is blood cells that still have the toll-like receptor 4 on them, but the vasculature is missing it. So we're not being able to answer this question here. And so we sought out to sort of make this toll-like receptor 4 complete knockout mouse that also then carried sickle cell. And so to do this, we had to do a series of breeding. And this is where Fuad's magic comes into place. And so we took our humanized Towns mouse and our knockout mouse, and we crossbred them to make a humanized, a mouse that had the humanized alpha and beta, but then also carried the toll-like receptor 4 knockout. We had to back cross those animals about 10 times to sort of get them back on the same genetic background and to decrease any kind of any leakiness of the human cassette carriage. Then once we got to the family breed 11, we were able to cross in the sickle cell mutation. We had to still then do some more back crossing in order to get a sickle mouse. And so finally, when we were able to finally get to these F12, F13, about 12% of our mice were the SS knockout mice. And so we were able to finally do experiments. So I showed data from stasis, and I should have probably put this slide earlier, but we use this dorsal skin fold chamber to view vasoocclusion. And so what we do is we implant a dorsal chamber on a mouse, and we do intravital microscopy to map out all the blood vessels. We can then add in our insult of choice, be it hemin, LPS, hypoxia reoxygenation, and then we can measure the occluded vessels using that intravascular microscopy going back to our mapping. And at the end of the experiment, we can sacrifice the animals for blood and tissue analysis. And so we finally achieved our knockout mouse, our toll-like receptor 4 knockout SS mice, and we were able to finally perform these dorsal skin fold models on them. And what we saw is that when we gave a bolus of hemin, that the mice that were knocked out for toll-like receptor 4 on both the vasculature and the endothelium were protected from stasis. When we gave LPS, they were also protected from stasis. And when we did hypoxia reoxygenation, they were protected for stasis, though that response wasn't quite as durable as it was with the LPS or hemin stimulus. So going back to a schematic of how toll-like receptor 4 works, separated up here in the middle. It signals on this MYD88 pathway, and it comes down to NF-kappa B. Well, NF-kappa B is one of those master transcription factors. It's involved in inflammatory genes, TNF, IL-1, IL-6. It's involved in ketokines, so CXCL2. CXCL8 is also known as IL-8, which we'll talk about later, but in mice it's CXCL8. And then the adhesion molecules, which many of us are familiar with. And so we wanted to know, if we knock out toll-like receptor 4, do we actually prevent the NF-kappa B signaling? So when we look at the mice that are sufficient in toll-like receptor 4 and we give them a stimulus, they have signaling, they get NF-kappa B signaling as we would expect. When we knock out the toll-like receptor 4, we see total absence of the signaling pathway. And this was done in liver tissue. We've also done similar blots in other organs, in particular the kidney. So we've really sort of knocked out this NF-kappa B response. And so we would expect to see a decrease in some of these downstream mediators. And so that's what we queried next. So I don't know how many of you have ever encountered Dr. Verslati or met him, but he can be a very excitable person at times, especially in our lab meetings. And so I remember we had our students, our summer students, and we've been helping them do these RNA analysis, and they were presenting in lab meeting, and they showed these data that, you know, there was no difference in our sickle cell mice that had toll-like receptor present or toll-like receptor deleted, and he was getting very anxious. Because we weren't seeing any difference in our chemokines, and this was just causing a lot of anxiety. And cooler heads prevailed, and we said, why don't we try giving them some hemin first, because maybe they're just not hemolyzing enough. And so we did. We went and gave hemin, and his blood pressure and everyone else's blood pressure came down, because what we see is that in the animals that had toll-like receptor 4, so these are toll-like receptor 4 knockouts or toll-like receptor 4 sufficient mice that have sickle cell disease, when you give a bolus of hemin, you see that the inflammatory cytokines do not get upregulated in the animals that are missing the toll-like receptor 4 gene. And in particular, this was IL-6 was included in this. Same thing with the pro-adhesive genes, so VCAM, ICAM, and E-selectin. We didn't see an upregulation of these genes or a downregulation of these genes at baseline. It was only when we gave hemin that we saw this protection, and that when you gave a mouse that was sufficient in toll-like receptor 4 that had sickle cell extra hemin, you had upregulation of that VCAM. When they were insufficient in toll-like receptor 4, that VCAM didn't get upregulated, the ICAM didn't get upregulated, and the E-selectin also didn't get upregulated. And we went and showed that the protein level, we're also seeing this. So this is again in livers. And when our livers are exposed to extra heme, if we have toll-like receptor 4, we upregulate those adhesion molecules. If we're missing toll-like receptor 4, we don't upregulate those adhesion molecules. So going back to cartoons, we now know that free heme binds to MD2 here. Downstream, it activates this MYD88, which then affects NF-kappa B phosphorylation. We get these adhesion molecules. But we hadn't really covered what goes on at the endothelium, the viable Plotty bodies, the von Willebrand factor and the P-selectin. And so we wanted to know, do we see the same degranulation effect? So we went to the organs. And so again, these are SS mice that are toll-like 4 sufficient here, and SS mice that are toll-like 4 deficient. And P-selectin's in green here, von Willebrand factors in red. And I think it's pretty here, or striking. One of the reasons I think crizanlizumab works is because we have P-selectin at the surface of this blood vessel here in this animal. And this is an absence of an extra heme bolus. In this animal, we're really not seeing any activation of anything, maybe a little touch of the VWF there. When we give an extra load of heme, though, that's when we really see, the presence of P-selectin and von Willebrand factor together, the green and the red are merged, so you see orange here at the surface of that blood vessel. But when the animal is insufficient in toll-like receptor 4, we don't see that response. So putting this together, we have loss of toll-like receptor 4 in hematopoietic and vascular cells in sickle cell disease leads to reduced heme-mediated pro-inflammatory and adhesive responses, and loss of toll-like receptor 4 in hematopoietic and vascular cells in sickle cell disease leads to reduced heme-mediated von Willebrand factor and P-selectin secretion or release. But we still hadn't answered the question. Is it the monocytes, the vasculature, the hematopoietic cells that are required? Or is it the endothelium? What is it? Is it the chicken and the egg? What's more important? And so we no longer had the issue of our chimeras not having toll-like receptor 4 on them. And so we could then go to a chimera system again, but this time be confident that we don't have toll-like receptor 4 in those monocytes and macrophages. So this time we took our SS toll-like receptor 4 mouse, so no toll-like receptor 4 present anywhere. And then we would cross it to AA mouse that had toll-like receptor 4 on the vasculature. So now we're really looking at, do you need to have toll-like receptor 4 on hematopoietic cells to then stimulate the vasculature, or does the vasculature have to have toll-like receptor 4? And what we saw is much like our wild type mice, a mouse that is lacking toll-like receptor 4 on hematopoietic cells and has sickle cell disease still has stasis. When we do the converse, when we have a sickle cell mouse that doesn't have any hematopoietic cells with toll-like receptor 4, and we take that bone marrow and put it into an animal that doesn't have any toll-like receptor 4 on the vasculature. So basically we recreate our SS toll-like receptor 4 mouse using a bone marrow chimera. We see that protection that we saw earlier in our experiments. But the comparison between these two is telling us that the difference there is that the vasculature has the toll-like receptor 4 ablation, not the blood cells. When we do this in another set of animals, so this time the vasculature doesn't have, and this is a wild type mouse, so this would be a SS toll-like receptor 4 plus, and the vasculature also has toll-like receptor 4, and we get that stasis response in response to heme bolus. But in this case, now we take our SS toll-like receptor 4 positive. So the hematopoietic cells have the toll-like receptor 4. So those monocytes can see heme, respond to it, and signal and create TNF-alpha and other cytokines. But the vasculature is missing that toll-like receptor 4. So when the vasculature sees that free heme, it's not going to signal through that MD2 interaction. And what we see is those animals are protected. So now we're able to say that in our system, it's the endothelial toll-like receptor 4 that's important for mediating the vaso-occlusive physiology. So that vasculature being bathed in that free heme activates those endothelial cells, leading to release of that von Willebrand factor and the P-selectin and upregulation of all these adhesion molecules, which can then perpetuate that vicious cycle. And so that's sort of our take home point. So putting this together, we've done a lot of work in mouse models, we've done some human cells, but how can we translate this? That's sort of the next hurdle. What is all this knowledge going to do for our patients sitting in front of us? And so what are some strategies to prevent this mediated endothelial activation? How can we put this together? So soluble MD2 is really actually a very understudied molecule, but it has been shown to be elevated in the plasma in various inflammatory conditions. Up until recently, we didn't know if it was involved in sickle cell disease, but things like inflammatory arthritis and other things that had been looked at. So we sort of looked at two different questions. Does soluble MD2 and heme interactions play a role in sickle cell disease? And do we have strategies to reduce this interaction and prevent that endothelial activation? So this paper, I think, came out a few weeks ago. I just call this the paper that killed my Christmas, but because we were revising this over Christmas. But what we did here was we looked in both, again, human and mouse, because we want to make sure that we're not just describing a very interesting thing in mouse models and nothing that would translate to humans. And what we see is that compared to AA controls, the SS individuals in mouse and have increased soluble MD2 in the plasma. First, we used Western blots to describe this, and then we eventually were able to find a ELISA-based assay, so we wouldn't have to run endless Westerns to look at our patient population. We see that there's an increase in soluble MD2 compared to AA controls. There's a little more spectrum with the MD2 release in our patients, which may reflect phenotypic differences in hemolysis. We weren't powered enough to look at that in this study. We know that the soluble MD2 binds heme, so looking at a heme aggros column, when we do a flag tag, we can pull down free heme. And then the sort band, which for people who are hardcore biochemists, what we see here is you have soluble MD2 alone in the light blue. And when you bind heme, you get a shift at a wavelength because heme proteins have a unique UV light scatter. So you get a shift with that sort band. And so that our patients with sickle cell disease have increased soluble MD2 and that this MD2 is potentially binding the heme. And then if we just take plasma from our sickle cell patients and we put it on endothelial cells. So we're not talking, Dr. Hebel has spent years isolating sickle blood outgrowth endothelial cells. We didn't pull any of those out. We just took human umbilical vein endothelial cells and we added the SS plasma and we saw an increase of CXCL8, which is one of those NF-kappa B dependent cytokines, or IL-8 as we'll call it in human. And what we also saw is that when we did sickle cell plasma and we added that toll-like receptor 4 inhibitor, we can block that release of that IL-8. So that's indicating that, again, that heme MD2, or in this case, the soluble MD2 heme interaction is signaling through toll-like receptor 4 on the endothelium to lead to IL-8 production. So to really sort of get at home on that, we did a strategy where we took sickle cell plasma and we used columns to deplete the MD2, soluble MD2, and then we treated the HEVEC cells. Or we took AA plasma and we spiked in soluble MD2 and put that on HEVEC cells and looked at IL-8. And what we see is that we have the SS plasma. When we deplete MD2, we get decreased IL-8 production. And when we took media that again had was AA or plasma, and we spiked in the MD2, we got activation of IL-8. And this time, if we added TAC242, we blocked that. Note with this experiment here, we didn't add any extra heme in there either. So this is the MD2 finding the unquenched heme within the plasma. So when we looked at other cell lines, so is this unique to HEVEC? We know that in particular lung pathology is important in sickle cell disease for a variety of reasons. So we did a human lung microvascular endothelial cells and we saw very similar findings of these. Not only IL-8, but VCAM and IL-6 are upregulated in the presence of sickle cell plasma. And then what's interesting is if you look at this I've mentioned hemopexin throughout and scattered throughout the talk here, but hemopexin has a higher affinity for that heme than MD2. And that's important because chemically we have a source of heme that's coming from hemolysis, and MD2 is out there and binding to it. And in most cases, we have hemopexin that's there as well, but in sickle cell disease, hemopexin is often, the production of hemopexin exceeds the amount of heme present in the body. And so that allows for this unmasking of this soluble MD2 toll-like receptor 4 reaction. And so what we did is we then added in, because it has a higher affinity for the heme, we took our sickle cell plasma and we added in hemopexin. And we show that we decrease the IL-8 production. And in fact, we can take IL-8, we can take AA plasma and add in the MD2. And if we add hemopexin, we interfere with that. Same thing here, we can take SS and add even more MD2, and it really gets an increase in that IL-8. But then again, if we add hemopexin, we quench that. So really hemopexin, in the kinetics that it interacts with heme at a higher affinity than MD2, can interfere with this interaction. Now, our group has shown in the past that if we take, again, these are stasis carbs from vaso-occlusion molecules. If we take animals and we give them a bolus of heme, they get stasis. And if we give hemopexin, we can reduce that stasis down to levels of control. And that hemopexin itself can decrease that NF-kappa B activation. And so putting this together, more recently, CSL Behring actually received orphan drug designation for a hemopexin-derived plasma product. And there's actually now a clinical trial in phase 1 development that we've gotten clearance to sort of start enrolling patients at the University of Minnesota and several other sites that will be looking at a hemopexin strategy to interfere with these toll-like receptor 4 interactions. So the estimated enrollment is going to be 24 participants, 4 per dose cohort, with 6 cohorts trying to just get a dose into place. It's a single infusion PK study. And they're really looking for adverse events with this. And anytime you give a plasma-derived product, you have to worry about that. So I think they're going to be paying close to concentration area in the curve and antibody development. The age is going to be adults, 18 to 60, and they either have to not be on hydroxyurea or be on a stable dose, not adjusting doses. They're not allowing for patients to be on novel therapy. So our patients on voxelotor or on CRIZ won't be able to enroll in the study. And they're fairly stringent with their exclusion criteria. So they really want steady state patients. And so any patient who's been in the hospital, had blood transfusions, is going to be excluded. And the one that made me a little sad when I was reading the study details is the vaccination within 30 days, because of course we're encouraging all of our patients to get vaccinated for COVID. So we'll have to discuss that maybe. And then the other one is history of anaphylactic type reactions. So this study, I think Alex is going to be the lead at our site, and we're very excited to sort of see this translation of this work that's been decades in the making, starting to actually hit the clinic or hit the bench. So to wrap this all up, vascular inflammation is important in sickle cell disease. We know now that heme binds to MD2 to activate toll-like receptor 4, and really the endothelium is critical for this reaction. We can disrupt this interaction with hemopexin, and that clinical trials to evaluate hemopexin are in the pipeline. So, and perhaps we'll have another tool to reach for our patients in the future. I put this into every talk I give. Our adult hematology group is really looking for anyone who's interested in benign hematology, sickle cell, or other disorders. We'd love to have you come and enjoy a nice Minnesota summer. I won't say anything about the winters. I like winter. I grew up in a much colder environment. So please take a look. And if you're interested, reach out to me or anyone else. And I'll take questions.
That was fantastic. Thank you so much. I've been fascinated by a lot of this for a long time. There are already some questions in the chat.
Mm-hmm.
So Dr. Pollack asks, would it be possible to formulate CSL 889 delivery via some other route besides IV?
And not too sure about that, in that with most of our plasma-derived products, if you do any hemophilia type work, most of the time it's going to be IV. One of the things that I'm sure that they're thinking of, or I'd love to see them thinking of, is can they pegylate or do something to make it have a longer half-life? But of course, we're still doing the dose finding on that. I know that in the past, one of the gene therapy constructs that I think I worked on in grad school, like when I was doing a lot of cloning, was actually a hemopexin overexpression construct. And hemopexin is made in the liver. So this is, thinking translationally, could we do something similar to we do for hemophilia and make an AAV overexpression construct in the livers to really help supply that extra hemopexin? And would that have adverse effects? So that's sort of some ideas that you could have there. So
there is a hemopexin study as well. So that's different from the CSL study is the CSL bearing study, right?
Yeah.
There's an interesting hemopexin study out of a pharma group I'm not as familiar with. We're going to be initiating here, called Vitafor. I don't know if you're familiar with that, but I think it's, I'll send you some information. I think it's fascinating. And I think it's interesting on a number of levels, not just for looking at the use of hemopexin and decreasing inflammation in crisis, but also in how it could possibly improve iron and iron overload.
Yeah, and I think that is one of those we get a lot of patients that are iron overloaded, and someone asked in the chat if we think heme could signal through other, we know that ROS is important. So I sort of minimized ROS in this talk because we weren't looking in that, but the ROS pathways in particular are very potent. And so I think in some of our patients that have iron overload, they're sort of a setup because they have ROS, a readily source of ROS right there all the time, and that source of extra iron. So that is sort of one of those things that I think it's going to be a balance of how we not only treat the free heme, and I think the free heme is really important in like vaso-occlusive crisis, but some of the chronic issues that we see in our patients might be driven by other non-free heme.
I absolutely agree. Another question about, does heme induce MD2 solubilization, from Dr. Patel?
So that, not entirely sure, because there's other things that it's sort of secreted. So it can be other things that can stimulate that. So is it, presence of TNF alpha or other things? We haven't really gotten into that. Again, it was sort of not a extremely well studied molecule. CD14's been studied a lot more. So.
And then another great question that wasn't great talk. Similarly, Dr. J. Pollock, we have two Dr. Pollocks just to make things confusing. Do you know if heme could signal via other pathways in addition to the MD2 TLR4 pathway?
Yeah, so again, that sort of that ROS related pathway is one that we know it can activate through. The TNF alpha effect, I think Bob had that. I think the Intercept paper finally came out. So heme can definitely, stimulate. I think there it's more of a, it hits the monocytes and may affect TNF alpha, but we can see that. And then of course, heme oxygenase is, was the first project I did. And we still go back to heme oxygenase, not doing it quite as much as we used to, but we know that pathway and the NRF2 pathway is important. And there are stuff that Dr. Versalotti and Dr. Belcher involved with separate from me, looking at NRF2 and BOC1 inhibitors to sort of get at that aspect of the heme signaling.
Interesting. Another question and comment, thank you for the very interesting talk. I may have missed it, but what was the evidence that endothelial TL4 was more important here than vascular smooth muscle TLR4?
So we really sort of ignored the vascular smooth muscle. So thanks for nailing that down. We have not done any smooth muscle cultures and looked at the toll-like receptor 4 on the smooth muscle. So that was sort of one oversight, but we know that based in our in vitro studies that the endothelium is important. And that's where we would expect more of our vasculature to have like the ICAM, the VCAM, the E-selectin, those type of things. Now, where the smooth muscle signaling could get interesting is in vasoconstriction. And so that would be an interesting avenue to look at. What could be colder than Minnesota? What I used to say when I lived in North Carolina was I grew up in the other state with North in it, and it is colder than Minnesota there. So North Dakota. Is heme activation of the endothelial inflammatory less potent compared to soluble MT2 bound to effects? Could MD2 be acting as a protective mechanism to sequester free heme? I think that it has to be there for a reason. I almost think that in the hierarchy of sort of protective things, we have a lot of, we have albumin, we have haptoglobin, which I didn't even cover. We have hemopexin. We have a lot of different things that we've invested making in our blood to protect us from free heme. And so soluble MD2 might be one of those ways, but it may also be one of those scavengers that's out there if you've already evaded everything else, and now you're binding to MD2. Now it's really time to signal and take this free heme on. So that's sort of my view of it is that it's there and it's probably getting released if the endothelium is a little irritated, but that when it's really released is when we get that system. So thinking about it in a coagulation pathway, we know that we get just a little bit of tissue factor activation, and then we sort of hit that amplification once we start getting factor 11 and we get that sonic boom that makes that clot. I think that's sort of what we're seeing with our heme here.
Interesting. And then Dr. Liebensberger asks, any changes in CBC with TL4SS knockouts?
No, that was also a very long few lab meetings where when we started characterizing these animals, there was really no change in any of that. And man, that was, why is this going on? What's happening? It was a long few lab meetings.
Another question, how much of TLR4 action is due to the NRF2 effect?
So that, again, we didn't really get into. That's a series of studies that stay tuned. We'll get to that.
That's awesome. So next steps, it sounds like you have a lot of work to do.
Yeah, we do. We're busy and we like it that way. We don't give us idle hands. It's tough when it's a sunny, nice day in Minnesota, but it's actually more fun then. So
I think this is fantastic and super important work. And I'm just glad you are here to present to us today and want to offer you our thanks again. And
there was one question about endothelium or nitric oxide. So we've not looked at ET1 and we've not done NOS. That would be, again, sort of interesting to do. In the original 2014 paper, we did do a series of the heme plus the ROS inhibitors, and the ROS inhibitors were potent in interfering with that effect. So sorry to interrupt. I wanted to make sure I answered.
I'm glad you got to it. I didn't even see it.
Yeah.
Anybody else want to unmute and ask your question? All right, well, thanks to everyone for joining, and we will see you next month, same time, same place. And thank you, Dr. Beckman, again for joining us and presenting such a great talk today.
Yep, thanks. Bye.
Bye.